Okay, so we're going to take our RNA and we're going to come up with a complementary strand. We're going to make this our mRNA. So this is one of our 3 forms of mRNA. So this is our first one, okay? So I am mRNA. Remember that our ribosomal RNA is what makes up the ribosomes. So that's what the nucleolus makes the ribosomes, remember? Okay, so let's get, so T, now T would pair with A, and G with C, and C with G, and G with, whoops, with C. A pairs with U, uracil, and T pairs with A. Okay, so I've got my strand of mRNA, and it is only single-stranded, and it is going to make its way out through the nuclear pores to the ribosome. So we'll just do our ribosome out here. Remember, that's made of our RNA, ribosomal RNA. Okay, and I'm going to go ahead and I'm going to put my strand. I'm just going to copy what I have here, okay? So A, C, G, C, U, and A. And keep in mind that this has, this would be a portion of the DNA, which is really important. So this is coding. This would be really determining, looking at your gene or gene expression, which is really focused on, you know, a certain section of traits. So this would be coding for a certain type of protein. So here we are out into the ribosome. And something to really pay attention with is looking at these in triplets, okay? And if you remember from biology, and if you don't, that's okay too, is that we call these triplets codons, okay? And so I purposely gave you only 6 of these so that you could, there we go, so that you can really focus on the whole idea of just two of these. So in triplets, so the codon, this is in triplets, okay? Now remember, to go from DNA to RNA, that was transcription. So let me back one step because I didn't write that. This is transcription. Okay? What we're doing is we're taking something and we're writing it in our own words. We're still talking English. But when we go out to the ribosome, now we're getting into the second part, which of course this is talking about, this is translation. Okay? Because now we're taking the A's and C's and G's, that sort of language, and we're changing it into our own protein language. So a completely different, we're translating, okay? Out and in at the ribosome, we are now going to form to our existing mRNA the complementary, or again, the opposite strands, okay, and our final type of RNA, which is called transfer RNA. So tRNA. Okay, so this is translation, transfer RNA. And on a transfer RNA, this is what we've got going on here. So say, for example, I'm just gonna pick some letters here. Okay, so I'm just gonna go CCC. Oh, CCG. Okay, CCG. So this is called a transfer RNA, this whole thing. Okay. Let me label the parts. This piece is the anticodon. This was the codon. This is the anticodon. This is what would match this. And on this anticodon, it is associated with a particular, what do you think AA stands for? You're right, amino acid. So depending on who it matches up, the amino acids then get put together in a chain. So in this case, let's see, who would pair with who? All right, so A pairs with T. Oh, I can't use T. I have to use U. All right, C pairs with G, and G pairs with C. UGC. C pairs with G, U pairs with A, and A pairs with U. Okay, and then what happens is that in between these, then what kind of bond forms? You're right, it is can you even see that? It is a hydrogen bond, remember, or we can call it, in proteins, we called it a peptide bond. Do you remember that? And this will continue on and on until you it depends, you will encounter a stop codon. And that signals the end of that protein. Now keep in mind, something really fascinating. Insulin is about 51 amino acids long. 51, that's crazy. Okay, so, and some of those, remember how I was telling you about making the protein and it all shapes? Some of those are hundreds long. So it's really kind of cool when you stop and think about it. Now, here's the other piece. How do we know which amino acids are which? This is where we use our fun amino acid wheel of fun. But something that you need to keep in mind, you need to write on this mRNA, because this wheel is based on the mRNA sequence. Now, I've argued with my genetics professor at college. Why is that? Because it's the tRNA that brings the amino acid, but it's the message that depends. So that's why you have to read it from the message. Okay, so in our case here, so let me zero in again this. So using our messenger RNA, okay, based off the messenger RNA. So let's see, ACG, okay, according to this, ACG. So here's how you do it, A, C, and G. Now notice these are all the same. That's interesting. Threonine, okay, so this would be threonine, okay, and I'm going to write based off mRNA, okay? So that's the only confusing part, that's where it's based off from. So there's that piece. I'm going to use highlighter. Okay, and let's see, this one is CUA. C-U-A, leucine. Leucine. And again, those would continue down the track until you get a stop codon. See, here's a stop here. Here's a stop here. Now, if you'll notice, we're going to come back to this again, but this is really important because there are some repeats in there. So we're going to talk about how come there might be some repeats and why. There's only about 20 amino acids. If you count these up with repeats, there's only 20 of these. And they actually are common amongst all living creatures, which is pretty fascinating. Okay, so there you go. That was your Lego building that you did in biology, but there is transcription in translation in a nutshell. And so the next question is going to be, taking these, what happens if you change up the letter or get a mutation? How does that switch up the amino acids? Thanks.